the data support your hypothesis, then you can write a paper. And if, for example, it's this one here, it's a pretty famous paper. First author and last author won the Nobel Prize 2 years ago because they found out how to make mRNA vaccines. And this is the famous study that came out in late 2020 when they had the first mRNA-based corona vaccine that really worked perfectly well based on the science that had been done before. So that will then earn you the applause and everybody is happy. You, of course, most of it, and that's great. So this is how we all want to have it. Unfortunately, from time to time, this might happen. The data just don't support your hypothesis. So what you then should do is go back to your sheet of paper and update the hypothesis and run the whole thing again. So these are the two main things. But unfortunately, there is a third possibility, and this is when something unexpected happens. And you're left with wondering, like, what the hell? I have totally no idea what that means. So how do you deal with this? So there's a bunch of possibilities. So sometimes, unfortunately, I believe too often something like this has happened. It's just put on the carpet. You don't want to talk about that anymore because it doesn't fit your expectations. Well, that's very dangerous to do. I give you an example. So what you see here are a number of frames, a video taken of the Space Shuttle Columbia back in 2003 when it was rushing to space. And can you actually see my mouse? Is it visible? Okay. Look at this little thing here that is exploding and making some dust. That is a piece of insulation foam that came off the big external tank and hit the corner of the wing of the space shuttle. Okay, so NASA had seen this movie of the space shuttle, as I said, January 16, and they investigated that, and January 31st said, well, no problem. And then 2 days later, the space shuttle returned from space to Earth, and very, very unfortunately, this is what happened. This is on February 1st, and here you can see how space shuttle is falling into pieces and is completely destroyed, and all 7 astronauts on board, of course, died. So you should not have said it's okay, because what they then did is a scientific investigation into the causes. And essentially, they did an experiment replaying this event of this little piece of foam hitting the corner of the space shuttle wing. And what you can see here, okay, so this piece of foam completely destroyed this insulation material of the wing, which was entirely unexpected, and rather than being no problem, it was a serious problem. So in the end of the day, the wing of the space shuttle was no longer protected, and therefore hot air could enter the wing and destroy the entire spaceship, leading to 7 deaths. So putting unexpected results under the carpet is not a good idea. You should not do this. Well, then what else do you do with unexpected results? Simply because you can't get them under your hypothesis, you don't know what to do with them. So you should not do putting it under the carpet is not a good one. If you don't know how to deal with them, too many people decide, well, I'm going to leave academia. I don't know how to do that. So I'm very happy that you young people are organizing yourselves to become great scientists, because we need you. Why do we need you? So here's this gentleman that's the current Secretary of Health in the United States government, and RFK is his name, and he has made a bunch of really great statements, like, for example, that vaccines cause autism or antidepressants lead to mass shootings and this ridiculous kind of things. I'm just mentioning a bunch of those. You can read them. This is, of course, entirely stupid, but we need clever and smart and well-educated scientists like you to counter argue and tell everyone else, no, please don't believe that, that's bullshit. So we need educated academics very, very much. So please don't leave university. So then there's space for one more, and that's actually what you should embrace. And that's what I call kiss of the muse. So this is the thinker and this is the muse, and he just gets a kiss. That means he is exposed to a new thought that he didn't plan to be exposed to. And a famous French scientist, his name is Louis Pasteur, came up with this quote here: chance only favors the prepared mind. So you can only discover that you have seen something magic when you have learned a lot and understand it, what you have seen. And this is making the difference of an average and a great scientist. And a lot of things we use today are actually based on such chance discoveries. So I'll give you some examples. Maybe one of the most famous ones is how Newton discovered gravity. And the story goes, here's this famous apple tree in Cambridge, in UK. And when he was a young man, he was sitting under this tree in a hot summer day, and suddenly an apple fell off the tree and hit him on his head. And rather than being annoyed because he was hit on the head by an apple, he thought, well, wow, why is this thing falling? And he came to the conclusion that the earth is attracting the apple like the apple is attracting the earth, and that led to the discovery and the formulation of the theory of gravitation and gravity, which is, of course, absolutely central to modern science. Second example, 1928, a researcher, his name is Alexander Fleming, was cultivating bacteria on those Petri dishes. Everything you see here as a spot are little bacteria. So he made those bacteria, and we are back in 1928. So it was very hard to get one of those plates because there was no big plastic or so. It was really a lot of hard work. And then he had some of those plates full of bacteria, stored them in his drawer, and went to vacation. And when he came back, he discovered that here a fungus was growing on this bacterial plate. So normally you would then have been annoyed by the fact that your bacteria have been contaminated with the fungus and would throw them away. But here's now the chance. What he also saw is, if you look at the area where this is a fungus, right? Very close to the fungus, there are no bacteria. They're only here, which is far away. So Alexander Fleming saw that around this fungus, there's something we call an inhibition area, which is because the fungus is making an antibiotic, penicillin. And so he discovered the first antibiotic based on this chance event of his plates becoming infected by a fungus. And that, of course, is a very fundamental discovery. Also won him the Nobel Prize and brought like any antibiotics to humans. So consider a life without antibiotics today. Last example is about something called cosmic background radiation. So this is an antenna that was used by those two gentlemen here, physicists Robert Wilson and Arno Penzias, and they were actually trying to measure some signal from a satellite in space. And in order to measure this very faint signal, they had to get away with all like disturbing additional signals, like from radios and whatnot. So they did a lot of things to clean all the not wanted like radiation away in order to just measure the radiation from the antenna. But very annoyingly, there was a radiation that was coming from all directions in space at a very defined wavelength of 7.2 centimeters, and they could not get away with that. It came in, day out, at night and in the morning, and there was no source on Earth that could explain this radiation. And ultimately turned out it's so-called background radiation of the space, and it is giving us a picture of the early space, like after Big Bang, and now we can map this cosmic background radiation. This is early data, and the most recent one is this one here, and it's a very important source of information on space. So this was also a chance discovery, and they also won a Nobel Prize by that. So chance is always happening, the key point is, what do you do with it? So I'd like to give you some basic rules of how to deal with unexpected events. So first, the data is the data. Unless you have made a very clear mistake in your measurement, if you have not done that, then take the data as they are. A famous physicist, Richard Feynman, I admire him very much, made this quote. So no matter how beautiful your theory is or how smart you are, if the theory doesn't agree with the experiment, it's wrong, very basically. So do not twist the data. The data are the data, very important. Second, and this is super important, make down as many notes of little things as you can think of. You never know what is explaining your data a week or a month later when you look at them. So little notes, like for example, I choose this vial to pipette stuff into my experiment and not the other vial, and then it turns out the vial, you confused the vials, and the vial with you were using was not containing the correct stuff, for example. So very good check of your experiments is super important. Third, do not remove unexpected data unless you have a very good reason to. If you know the machine wasn't working, you had I don't know, an interruption of power supply, whatever, and you can see that, then it's of course okay to remove bad data. But if you cannot explain that, then leave it, and you need to work with it. And maybe that's your discovery. Another one, the moment you become, let's say, group leaders and have PhDs you are working with, and you are supervising the PhDs, a very important one is this one here. Don't yell at your subordinates. If they come to you with unexpected results, listen to them. Maybe also encourage them to tell all their stories. Tell them it's not forbidden to show crazy data. Maybe the crazy data is just this chance event. And the final one here, I know that's easier said than done, but please resist the pressure to publish if you can. Look at your data, take your time. A good story will take time to evolve before it's ready to go. So this is more general approach I wanted to share with you about unexpected events. And now to give you a practical example, I would like to share a story with you from my own lab. It's also a nice story because Sushmita, who is among you, was a very central figure in that story. So it's about a serious disease, stroke. You all know what stroke is, and stroke is still a main killer. That's data from 2021. Today, stroke will be on position 2, very likely. Again, because COVID-19 is no longer a problem. Stroke will be probably number 2 killer in the world. So it's very serious, according to WHO. So importantly, stroke affects multiple organs. Of course, it starts in the brain with some clogging of a blood vessel that is then killing neurons, but it also has very massive effects on other peripheral organs, especially also the gut. So you will suffer from inflammation of the gut, dysbiosis, gut barrier leakage, and ultimately, in the long run, people that suffer from stroke will have suppressed immunity for quite a while, and that can lead to post-stroke mortality. So people that have suffered a stroke can get an infectious disease very easily that can kill them, which they would normally easily fend off. So this can happen. And the reasons for that are plenty, but one important one is happening really in the gut itself. So because of this leakage of the gut, etc., many things, the bacteria in the gut, which altogether are termed microbiota, are very heavily changed. And this has a big impact. So because of this fact that stroke was affecting the gastrointestinal tract, we were looking at the immune system of the gastrointestinal tract with high attention at the time. So the immune system of the gastrointestinal tract, to a large extent, is based on a structure termed Peyer's patches. Now, Peyer's patches are, if you want, lymph nodes in the small intestine. And inside of those Peyer's patches, there is a type of immune cell termed plasma cell. This is a B cell variant that produces a lot of antibody. And the antibody is specific because it's termed IgA. This is antibody that can cross the gut epithelium and go into the gut lumen and then bind to the bacteria of the microbiome. Now, under healthy conditions, this binding of IgA from Pius patches to bacteria of the microbiome is producing something like stable situation. Normally, you will not be killed by your bacteria in the gut. You actually need them. But it only works because you have IgA, and that is coming from plasma cells in the Pius patches. So why am I telling you this? Because of this connection, we were looking in detail at those Pius patches. Now, if you take the gastrointestinal tract of a mouse, this is the stomach, and this is here the colon, and along the small intestine, you will find a lot of those Pius patches. For example, here, and you can look at this. This is a Pius patch here. That's about the size, 2 millimeters maybe. And you can compare it to the head of a needle, right? So this is the size of a healthy Pius patch. Then you can do some imaging, and you will be able to show it's full of those plasma cells, which you can label with this molecule term CD19. You can also make a three-dimensional reconstruction. Then you will see a beautiful Pius patch full of those yellow things. They are, of course, yellow because we stained them yellow, and these are the B cells. So the Pius patches have a ton of those B cells. Here you can see them, and that's how it should normally be. Now, the Pius patches in a mouse, you can see with your naked eye. You don't need a microscope. They are big enough to see them. So then one day, it was really like that in the lab. A student in the lab, Alexander, came and said, I can't find the Pius patches. So I said, what do you mean you can't find them? Yeah, I can't see them. And so I said, what did you do to this poor mouse? And I said, yeah, it's suffering from stroke. So I said, are you telling me you can't see the Pius patches in the mouse that has suffered from stroke? And he said, yes, that's what I want to tell you. And does anyone know this? And I said, no, no one knows this. And so I said, wow, so we should better follow up on this. So looking at that, here's a stroke Pius patch. You can see the size difference. It's very, very dramatic. So this is a moment of discovery. This is the chance event. It's Alexander coming into the lab and saying, I can't find the Pius patches. Now, what I could have been doing as boss, I could shout it at him and say, hey, go back and look better, you fool. You just didn't look at this well enough. That would have been a very bad idea, because then we would have all missed it. And then I instead said, wow, this is very interesting. Let's look into them in more detail. So that moment, and that's now 6 years ago, when Alexander came in and said, I can't find the Pius patches. That's the moment of discovery. That's the kiss of the muse moment. And we luckily understood the message nature was giving us, and we were following up on this. And here you can see some data now. You can see that 3 days after stroke, really, that's the red dots here. The number of Pius patches you can isolate from a mouse is much smaller than in a controlled situation. Then we did some imaging. This is a Pius patch in microscopy. So the plasma cells are green. In between are some other cells termed T cells, and then the rest of the structure here is just the gut. You can see how a pretty Pius patch looks like if it's all good. This is the Pius patches along a gastrointestinal tract of a mouse, of a healthy mouse. And this is a mouse that is suffering from stroke. And especially here at ileum, for example, you can see the size difference. That's why Alexander couldn't see them anymore. And this is the key discovery. So they are just melting away like ice in the sun because of the stroke event. So I told you about IgA. So if we then said, if in the Pius patches, plasma cells are present that make IgA, and now all the patches have shrunken, there should be less IgA, right? So we went to our clinical colleagues and said, can we please have blood from stroke patients and measure IgA in it? And that's what we did, and see the difference. So a day after stroke, or up to 3 days after stroke, your plasma IgA is dropping massively. The problem is, of course, you can't study the pathomechanism in human patients. So we made a mouse model where we have a mouse that is suffering from stroke, and we see the same thing: loss of IgA both in the plasma of the blood and also in the feces directly. And then you can do measurements, and you can see the same is happening actually in another very serious disease, which is heart attack. We see the exact same thing also after heart attack. The pious patches are melting. Same again in a mouse model of heart attack. And again, in patients, heart attack patients also suffer from this dramatic loss of IgA. So at this moment, we could say, well, obviously, if for some reason we have a sterile tissue necrosis in the brain or in the heart muscle, we lose pious patches. And that would be a pretty good moment now to say, well, this is a great hypothesis. Now it's grant writing time. Sit down and write a grant about this. So the data we have summed up to this moment, pious patches shrink, IgA goes down, both in people and in animals, and we see the two things both in sterile inflammation of the brain as well as the heart. That in itself would have made good data, preliminary data, to then write a grant and say we need to investigate that further. And that means for a good grant, you need a good hypothesis. You need to show that this is not yet known. This is very relevant, ideally. So the human data are very important. And we have some very good ideas of how to further study that. Okay, so honestly, we didn't write a grant. We just kept working because we had some money. We didn't need a grant at the moment. So let me sum up the story. I'll try to be a little bit faster, maybe not all the details, to give you the main story behind that really unfolded. So again, we see here the B cell loss in the patches. We also see that T cells in the patches are lost. This can be brought down to plasma cells. Plasma cells are lost. That's why you don't have IgA. And also mature plasma cells are just going away. And you can also show, that's the gray bar here, that the plasma cells in pious patches are on the way to die. This is termed apoptosis. You can measure that. And very importantly, the situation is lasting very long. So this is where the stroke is hitting the system. At 3 days, all the plasma cells are, or most of them are gone in the pious patches, and it takes more than 3 weeks for the mouse to recover. And that pretty well explains why people suffer from post-stroke immune suppression so long. So then why does that happen? What we then did is we further investigated that, and we found out if you look into the blood of those animals, what you see is a big increase in free DNA. You can just measure there's a lot of free DNA in the blood of those mice after stroke. And importantly, the free DNA is not there just like that. It has a function, because this is what I just showed you. We lose B cells after stroke, but this free DNA can be destroyed with DNA. So we injected DNase, a DNA chopping enzyme, into the blood vessel of mice. And if we do that, then we do not lose the B cells. So the DNA is not just there, it also has a function. It somehow kills B cells, the DNA. Same for T cells, by the way. And DNase can block that. So then where does that DNA actually come from? That was a big question. And a lot of people in the field thought, well, I have a stroke, so a lot of neurons will die. Neurons contain a nucleus full of DNA. It's coming from the neurons. I can tell you this is wrong. You have to think something completely different. And it helps that I'm an immunologist. I'm not so much a neurobiologist. I'm thinking about immune cells. So one very important immune cell that by definition can release its nuclear DNA is termed neutrophils. Neutrophils are the most frequent immune cells in the circulation, and they can release DNA as a defense mechanism. Here you can see an experiment. All the gray things are neutrophils, and this pinkish thing is a fungus that is producing a hypha. And look at this neutrophil here in the front. It's swelling, and it's becoming redder and redder. I can explain to you in a second why this is. And suddenly this happens. So the neutrophil ruptures and everything becomes intensively red. Why does it become red? Because in the supernatant of this experiment, we have propylium iodide. This is a dye that when binding to DNA becomes very, very red. So by this experiment, we can show, whoa, this neutrophil was exploding and all the DNA of its nucleus is now lying around here. And this is the defense mechanism that has been described some 20 years ago, that neutrophils use their nucleus to throw out the DNA, and the DNA itself is toxic to pathogens. Here you can see how that looks in high resolution. So these fibers here are made of DNA, and these, for example, are the fungal spores. So the DNA really sticks to the fungal spores. The key point is this DNA from such neutrophil explosions can be diagnosed because it is decorated with histones, and those histones also have a specific post-translational modification, which you can measure by an antibody. So what we then did is we went back to this experiment, say, if it's from those DNA nets, it should have histones where an amino acid is translationally modified into citrulline. So especially H3 histones can become citrullinated, and this can be measured with a specific antibody that recognizes CDH3. So again, this is a healthy mouse, and this is after stroke. And now we can measure not only DNA, we can measure CDH3 DNA. And now we know, whoa, the DNA after stroke in the blood is actually from neutrophils. We can, of course, chop this down with DNAs, but what we now do, we inject an antibody that depletes neutrophils. So now we have a mouse that suffers from stroke, but there's no neutrophils in this mouse as well. And boom, you don't see any of this DNA anymore. So the source of the extracellular DNA after stroke is very clearly neutrophils. And so then the next question, and here's where Sushmita comes in very importantly, is can we show that those neutrophils in a stroke mouse are activated? So we made the following experiment. We induced stroke in a mouse. I removed blood, isolated the neutrophils, and gave it to Sushmita so she could do mass spectrometry of the neutrophils. And sure enough, she could show that brain injury neutrophils are very, very different from healthy or from controlled neutrophils. And cut a long story short, those guys are very massively activated also towards the function of making those nets. And indeed, you can show that, again, Pius patch cell number goes down and then in a normal stroke. But if we reduce neutrophils by injection of an antibody, this is not happening. And here we have made a mouse that by genetic tricks doesn't even have neutrophils. And also here, B cells are not lost. So neutrophils are really important. And then you can also look at the Pius patches, and you can see while in a normal mouse with neutrophils, the Pius patch shrink. A mouse that has no neutrophils, the Pius patches remain stable. This is just showing all the things I just showed you. So you would normally lose B cells in Pius patches unless you inject DNAs, unless you remove neutrophils by an antibody, or you make a mouse by genetic tricks that has no neutrophils. So neutrophils are very important in this process. So then next question, how do they do this? Why do nets kill lymphocytes? So first we checked in a cell culture experiment, we isolated B cells from Pius patches and exposed them to plasma from a stroke mouse or from a healthy mouse and plus minus DNAs. And what you can see here is B cells become, caspase is a marker of apoptosis. When you take plasma cells from a Pius patch, expose it to stroke plasma, then they become caspase 3-7 positive. If you chop down the DNA with DNAs, it doesn't happen. Same for T cells from a Pius patch. So the DNA is directly toxic, but that's not all of it. What we can also show is that the B cells in those Pius patches actually suffer from a complete metabolic shutdown. This is looking at the B cells now, not with proteomics, but with RNA sequencing. Again, B cells from a stroke Pius patch are very different from B cells from a control environment. And if you do the analysis, you will see this is all metabolic activity in B cells, and that's all lost in Pius patch B cells of a stroke mouse. So somehow the B cells metabolically shut down. And how are nets inducing this? Completely unclear to us. So for this, we can again have a look at the Pius patches. This is a normal Pius patch. Important thing is, you don't see 3, 4, 5 cells here. You see about 100,000 cells here because this is a low resolution. This green spot here contains tens of thousands of Pius patches, and this one here as well. So what all those Pius patch cells need in order to live is a good blood supply, right? So what you can then do by microscopy, you can show the entire blood vessel tree of such a Pius patch, which is shown here. And what we then did is we took a an image of those blood vessels of a pious patch in a control mouse and a stroke mouse. And a stroke mouse we had been treating with DNAs. You see two colors here. I hope none of you guys is red-green blind, otherwise I have a problem now. You see red is the blood vessels and green are thrombi. So let me show you the data. You can see that in the stroke brain, there's a lot of green. That means a lot of microthrombi in the blood vessels. They are all full of that. And if you inject DNAs, there's nothing. So meaning that the net formation is causing microthrombosis, so it completely disrupts the perfusion of all those hundreds of thousands of B cells, and therefore they simply just suffocate, they die. And you can see the extent of those thrombi again in this picture here. It's really all full of those little thrombi, and they are produced because of the nets. And therefore, if we digest the DNA, this is no longer happening. How much time do we have left? Are we good, or are we is already tight?
You still have a few minutes.
I can't hear you. I'm sorry. Oh, wait. Say again.
You still have a few minutes.
Okay, cool. I'll do speed up a bit. Maybe I leave out one part. I'll share you from this. This is too much. We can stop the net release and then we hear it. But the key question may be this one, and then I can finish and we can have questions. What about humans? So we now measure net DNA in humans. And we see again net DNA, now SITH3 in humans after stroke. And importantly, the more nets you have, the less IgA you have. So it's a clear correlation. Same can be shown for human heart attack patients. So the question now was, can that actually be treated in the clinics? And for this, we teamed up with a French group in Paris around Michael Mazzighi, because they were studying stroke patients who received also DNAs from them for a completely different reason. It's not important at the moment why this reason is. Point is, we could get blood from them of stroke patients, human stroke patients, after admission to the hospital and after treatment, either with standard of care, which is called TPA. This is a treatment to dissolve the little thrombi, or DNAs plus TPA. So here's the data. First, you can see this, the pink circles are DNAs plus TPA, and you can see that SITH3 concentration in the blood of the patient is going down. So net dissolution with DNAs in humans is working. But what about IgA? So these are the normal patients that don't get DNAs, and look, after 24 hours, many of them have lost a lot of their IgA in the circulation. Some of them even 30–40%. But people that receive DNAs don't lose IgA. So it's completely transferable to humans, what we've seen. So to summarize here, it started all from Alexander telling me I can't find the pious patches. And luckily enough, I listened to Alexander, and we got the kiss of the muse right, and we followed up on that. And this ended up in a nice paper we published last 2 years ago in Nature Cardiovascular Research. So, summing up, it happens that you see unexpected things, but please don't throw them away, embrace them. Be aware that they might discover or contain the next big thing. Also very important, if you have PhD students or masters, bachelor, whatever, even student helpers, listen to them. If they tell you funny stories, there might be a big discovery behind. So this is the people involved, some of the pictures, our funding, and here I would be then stopping and would be happy to take big questions. Maybe I can unshare so we can see each other, right?
Yeah,
good. Here I am.
Thank you very much for this very round and inspiring story. I hope it's a great example for people, and you are as impressed as I am. We have quite a few questions in the chat.
Go ahead.
On different topics. So let's maybe first start with the ones that were closer to your talk. There was one question saying, how do you think the pressure to publish is affecting the way people do science?
Yeah, it is of course clear, because without a paper you cannot normally defend a PhD, or if you defend, it's going to be a not great mark, and that means you will not get a good postdoc, maybe not a postdoc at all. So I do see the point. We cannot say it's not relevant. Still, we should try to resist as long as possible. And for example, I'm much more into having this one good paper rather than 3, 4 average or low impact papers. Therefore, I'm not a fan of this counting numbers of papers. So I do not believe the PhD candidate with 3 papers is a better researcher than the other candidate which has one really serious paper. So, but I do see the pressure to publish. I don't have a magic trick how to completely avoid it. But if you can, by any means, try to follow up a story more deeply and write this one serious paper rather than writing 10 irrelevant ones, all of them with small incremental increase in knowledge. I hope that answers the question somehow.
Yeah, there's one question that's actually related to this one. Someone was asking, is there any way to improve the probability of these chance events appearing in your research?
No. What you can do is look at interesting questions, and then just do good experiments. Make sure that your experimental approach is serious, and you have done everything you can to control that your experiment is technically sound. So before you do a real experiment, really train yourselves and make sure that you have everything very well under control so that you can make sure technically my experiment was done properly. And then, yeah, well, it can happen. But I wouldn't know of any way of how to increase the frequency of chance events. This is not, I would not know how that would be possible. What I can also probably tell you is know your field, read a lot of things, because only if you have experienced a lot of things or read paper here and there, you can come to the conclusion, whoa, this is something no one has actually realized. I haven't read that anywhere. This is interesting. For example, when Alexander came to me and said, I can't find the Pius page, one of the things I asked him was, wow, is that known? And he said, no, that's not known. So here we go.
So you showed in your presentation at some point that would be enough data to write together a grant, that you had a hypothesis. So how much data do you think is needed before you start writing a grant?
Very important, very good question. So essentially when writing a grant, yeah, 2 things can happen. It's getting funded or it's not getting funded. If it's getting funded, you did all right. If it's not getting funded, this can have several reasons. And regarding the data, it can have 2 reasons, and they are essentially mutually exclusive, so a bit strange. Reason 1 can be the data you show are not convincing to support the hypothesis. We don't believe you, we don't fund you. A second crazy thing that can happen is the data you show are so much, what do you want to do research anymore? We don't fund you. You already know everything. So, and this balance, I believe, in my experience, is very difficult. I've seen both in reviews I got for my grants that people told me, well, you know everything. What do you want to do research? We don't fund you. And also, hey, we are not convinced. There's so many open questions. We don't yet believe your hypothesis. So you can, and probably a good experiment you can do is take your hypothesis, take the data you have to support your hypothesis and show this to people, to your colleagues, and expose them to what you have and what you would like to then follow up on in terms of a project, and just hear their opinion. What do you think? Is that convincing, or do you not believe yet? Or so. This is probably an experiment you can do.
Okay, thanks. There's another question that in part you already answered, but I'm going to include it for completeness. Someone asks, What are strategies or what strategies have you found most effective for convincing reviewers that a discovery-driven project is worth funding despite its inherent uncertainty?
I have no magic trick. Sometimes it works and sometimes it doesn't. Honestly, I have to say, it's getting more and more complicated to come up with some really unexpected hypothesis and get that funded, because funding agencies, reviewers, and so much more like a clearly laid out path where there's little chance of failure, which I honestly hate, because that's now not how basic science should work. Basic science should follow up on the crazy ideas. And this is where interesting discovery is hidden, not in some stuff there where it's pretty clear how you do it and that the results will fit and then move us all a little bit forward. So I have no magic trick. Sometimes the discovery, the data, the preliminary information are so good and so convincing that people buy in, but there's no guarantee. You have to try it. Your writing needs to be convincing. So it's also a good idea to let other people read your grant and say, would you fund me for that?
There's actually a question in the chat that relates to that. Someone was asking, your ideas are challenged by a coworker. How do you put your ego aside and objectively evaluate their arguments?
Beautiful one. It's the best a coworker can do to you. Hard critique. The one thing the coworker has to do, he should say it probably politely. And you as a receiver of critique, please separate critique for your ideas or about your ideas from critique for you as a person. Completely different story. So the best thing a colleague can do to you is give you tough critique and really give you a hard time, because what that is doing to you is it helps you make a better grant. And therefore, this is a nice service that someone can do. And I don't talk about ego. This is irrelevant. Forget your ego. Someone who's really giving you a tough critique, but not a stupid critique, of course. A tough critique based on like testable criteria. That's the greatest thing you can do. I know, I mean, I've been asked by people whether I could listen to their things and give my thoughts. And sometimes they are, let's say, mad at me because I'm doing this very tough. If I think an idea is principally great, but there's problems here and there, I'm saying that very clearly. And this is not to insult people. This is trying to help make a grant better, or honing an idea so that it becomes more mature and better.
All right, thank you. There's some more questions on what happens when you find something unexpected. Like, for example, how to recognize when you are over-analyzing the data, and you can be when can you be sure that the data is analyzed in the best possible way to put it forward without any bias?
Good one. So easy thing is, if you compare condition A with condition B, blind yourselves to what is what. So if, for example, we were looking at the DNA in the blood, and we knew that the blood here was coming from stroke and the blood here in control was control, believe me or not, this is kind of shading your view on the data, because you already know there should be DNA there, and then you will somehow see it. Therefore, blind yourselves towards what you're analyzing. That's a very important thing to do. What you can also do is, I guess, again, expose your ideas, your analysis to experts in your laboratory, in your university, or somehow in your network, or ask someone whether he, she knows someone who knows and who can criticize. This is kind of thing. And of course, today, I mean, we can all use AI and ask, is anything of that known? or so. You can essentially prompt an AI and say, what would you say if you would see this and this? This is absolutely doable. You can. This is the possibility we only have since 3-4 years, which you should certainly use.
And so, what if you find this chance to chase something unexpected, but it would really delay getting your PhD finished? Would you still chase it? How do you evaluate whether it's worth it or not?
It's not delaying your PhD. It's making an average. All of you have a good boss, and he or she actually sees it and says, "Whoa, whoa, whoa, cool, go for that." Let me put it this way: if you, I don't know, if you're hiking a mountain and suddenly discover a grain of gold, I mean, you couldn't plan it, but you should probably pick up this grain of gold just because it's lying there, not because you plan to find a grain of gold. And I think one important thing is, if you have been a lucky person and had this chance encounter and you were kissed by the muse, well, this is a gift, a present that the muse made to you. Then what you have to do is follow up on this. If you don't, well, then don't. But you will lose a moment. So you need to find a way of. It's not happening so often, and therefore, if you're the lucky person to whom it happens, follow it.
Thank you. There's another question coming back to the use of AI, which you just mentioned. Talking about AI, how are grant committees adapting to the increases in use of AI?
It's a fact of life. It's not going to go away. So one example is the biggest German funding agency, German Research Foundation, is officially allowing AI also for reviewers. You have to state you have been using AI. For grant writing, same thing. People will use it. It's ridiculous to refuse picking this up. This is there. Everybody uses it. Use it. My personal experiment with AI is it is hallucinating and it's telling you the biggest crap from time to time. So it can invent DOIs and papers that don't exist. So it's definitely your requirement to make sure that everything you pick up from AI is also correct. Don't just believe because ChatGPT is telling you this and this has been found out, that this and this has been found out. First thing you do is, if you ask ChatGPT, please give me a reference for that, and it gives you a reference, you pick it up and say, wow, this DOI is not even existing. It happened to me all the time. And then you can probably use another AI, like, I don't know, Claude or so, Perplexity, what do I know, and just double check. But let's face the fact that AI is there. It's not going to go away. So we cannot not use it. It's impossible.
Okay, then let me close with a very different question because I think we've covered this field pretty well. How do you actually become part of a grant reviewing committee?
Yeah, that's I can tell you a story how it happened to me. So I was this a young professor at a large department with a big professor, and the big professor was invited by, in my case, the German Research Foundation to make a big reviewing activity where he had to look at more than one grant. It was even with traveling and going there and doing this. And he asked me whether I wanted to join. And before that, he had talked to the funding agency whether they would be okay if a junior person would just join. And I said, oh, yes, I would very much like to join. I wanted to learn how to do that. And that's how I went into it. So I guess if you're very junior, the chances you are directly asked by a funding agency whether you want to be part of a reviewing committee are not very big. The bigger chance is that you have a big, let's say, a good senior person above you that realizes you want to do that and therefore invites you into such chances. What you can do is certainly reach out to your senior and say, look, whenever there's a chance, I would like to do that. And for example, before you review grants, it's more likely you're going to review papers. The lab directors get a ton of requests from the journals to review their papers, and they cannot do all of that. So therefore, in my particular case, I'm rejecting, I would say, 9 out of 10 requests to review a paper simply because I don't have the time. But what I do is ask my coworkers whether any one of them would be interested in reviewing this. And this is how you essentially get into it. And I can only advise you, do this reviewing thing. It's very important. It's teaching you a lot. It's teaching you about new ideas, research that other people do. It's teaching you about how a good and also a bad grant or paper is written. I mean, you review, and if you then say, well, this was kind of strange. I didn't understand what they wanted to do. I thought the methods were not suitable. The preliminary data didn't convince me. And then there's this other project which was extremely well written. I know very well what they want to do. I consider this super important, and the data are just mind-blowing. So they should get all the money. And of course, then looking at this cool grant, at this cool paper, you can then read that and say, okay, why is this a good paper for myself? And can I learn from this good paper how I'm writing my next paper? So for this, it's a very helpful and important exercise to do reviewing. Yeah.
All right. Then I think in the interest of time, let's wrap it up. Thank you very much for that super inspiring story. And I hope we all go away a little bit more open-minded towards our science and results. So yeah, it's been a pleasure. And also thank you everyone for attending. And if you have any more questions, also feel free to reach out to us. We may be able to,
Yeah, you can find my emails. If you want to talk to me directly, just send me a mail. So thanks very much for having me. I enjoyed.
Thank you very much for coming today.
Thank you.
Bye, everyone.
Take care. Bye-bye.